Hello again and welcome. I'm Laura Peterson. This At the U at the Center series comes to you from the Osher Lifelong Learning Institute at the University of Minnesota. OLLI is a learning community for people 50 plus who are curious, lifelong learners. We offer hundreds of non-credit courses a year, plus other ways to get involved, like special interest groups and volunteer opportunities. I'm a volunteer too. I want to extend a warm welcome to our OLLI members who are on this webinar, as well as our guests. Today's presentation is titled The Evolution of Cancer Therapy with Dr. Christopher Pennell. Dr. Pennell is Associate Professor in the Department of Laboratory Medicine and Pathology at the University of Minnesota. His research explores novel strategies for immunotherapy, drawing on the immune system's inherent specificity as part of a personalized medicine approach for treating cancer. Dr. Pennell also serves as the Masonic Cancer Center's Associate Director of Education, and he co-directs the Student Outreach Program for the Institute for Engineering and Medicine at the U. Dr. Pennell, I will turn things over to you.
Thank you, Laura. I appreciate the invitation, and I'm looking forward to the questions. So I will see if I can share my screen, which I can. So here we go, the evolution of cancer therapy. I'm going to start with a story. This is a picture of Emily Whitehead taken on her 5th birthday. 2 weeks after this picture was taken, Emily was diagnosed with a type of leukemia, which is a white blood cell cancer. Her parents were obviously devastated by the diagnosis, but were heartened by the prognosis because this was about 11 years ago. The long-term prospects for Emily's survival were great. They were about 90% for children with this type of leukemia. If she had been diagnosed in the 1960s, her chances would have been about 10% for long-term survival. So between 1960 and 2010, in those ensuing 50 years, research has increased the life expectancy for children with this type of acute lymphocytic leukemia from 10% to 90%. So her parents were encouraged by that. They then went through the standard chemotherapy regimen, and Emily's disease was put into remission. Unfortunately, that was relatively short-lived. It only lasted for about 16 months. And then her disease came back with a vengeance, and it was now resistant to the previous cocktail of therapeutics that had put it into remission. And so she was given higher dose, more intensive therapies, and those also failed. So by the time Emily was finished with all these, she really had no course left, and she was only given weeks to live. And I'll come back and finish Emily's story later on, but obviously it has a happy ending. So to start with my discussion of cancer, I thought I'd start with some basic facts. We care about cancer because 40% of us will get it. 1 out of every 2 men and 1 out of every 3 women in the United States will develop cancer in his or her lifetime. So if you yourself do not develop cancer, someone you know and love will. And this is why we care about it. Cancer's been with us forever. So some of you may recognize Gary Larson's cartoon on the left here. The real reason dinosaurs became extinct was because they smoked cigarettes. But the point is that we know from fossil records that dinosaurs got cancer. So this is a fossilized thigh bone from one of these dinosaurs. And that area on the right is a hollowed out area that's consistent with the tumor having been there. We also know from recorded history that cancer has been with us for as long as we have recorded history. So this is a picture of a bust of Atossa. She was the queen of Persia back in the 400 or 500 BC's era. And we know from records that have survived that she had a form of breast cancer that was incredibly painful. She had a physician slave at the time who was from Greece, and she asked him to perform a mastectomy to relieve her of the pain. She survived the surgery, and when she was recovered, she was disease-free and was so happy that she was disease-free that she said to her to her husband, who was the king of Persia at the time, Darius, "Look, I want to free my slave. I want to give him some lands in Greece to thank him for doing this." And her husband said, "That's great idea. We don't own Greece." And she said, "Go get Greece." So that was part of the reasons for the Greco-Persian War. that was made famous by the stand of the 300 Spartans at Thermopylae. So we know cancer's been with us for as long as we have recorded history. Unfortunately, it will be with us forever because it's an inherent part of our biology. Cancer arises from mutations that we'll discuss in a little bit, and mutations are the substrate of evolution. So if an individual has a mutation in her eggs or his sperm, and that mutation is passed on or inherited by the progeny, and that mutation confers some selective advantage to the progeny that allows them to reproduce at a higher rate, let's say, or have their progeny, their children, survive longer, then eventually that mutation will become the dominant one in the population. So this is the basis for evolution. Mutations in egg or sperm cells that can be inherited. In contrast, mutations that occur in non-heritable tissues, such as breast, these are called non-germline. So germline tissues are eggs and sperm, non-germline are everything else. These are not inherited. And if the mutation occurs in a particular gene at a particular time, it can lead to a malignancy like cancer. So these mutations occur naturally, and they are the substrate for evolution, but they can also cause cancer. So what is cancer? It's an umbrella term used to describe over 200 different diseases. At the last I looked, we had identified about 213 different cell types in our body. That number keeps increasing every year, but not at as great a rate as it did earlier. But each cell in our body has the potential to become malignant. And so the term cancer does not describe a single disease, but rather over 200 different diseases. These geometric shapes represent different tissues in our body. So the pentagram would represent white blood cells. So there are different types of white blood cells depicted by these different colored pentagrams: myeloid cells, erythroid cells, white blood cells, lymphocytes. And then within a given type of cell, there are different types of cancers that can arise. So in terms of leukemia, there's myeloid leukemia, B-cell acute lymphocytic leukemia, a variety of other leukemias. And then within one tumor itself, there is genetic heterogeneity or genetic variants that arise. So most tumors arise from a single progenitor cell that went rogue, that acquired the mutations necessary for it to become malignant. And that one cell divides and divides and divides such that it has billions of progeny. This process, though, is error prone, and so subtle differences can arise within one of these populations of cells. So now you're not dealing with a single disease, but you're dealing with multiple subsets of related but distinct diseases. And then to kick it all off, tumor cells modify the environment in which they grow. So they modify their neighborhood, if you will, or for example, I had an undergraduate in my lab one time and I was describing this and she said, so let's say I rent an apartment and there's furniture in there from the previous tenants and I don't like it and I want to move it around or get rid of it. Is that what you're talking about? And I said, yeah, that's exactly what I'm talking about. So cancer cells have the ability to modify the healthy cells in their local environment such that those healthy cells promote the growth of the cancer cells. And so all of this leads to an incredibly complex series of events. That is why cancer is still vexing us today. So when someone asks, when are you going to come up with a cure for cancer? My response is, well, when are we going to come up with a cure for infections? And then the answer is, well, there's different types of infections because there are different viruses and bacteria. And I say that's exactly my point with cancer. The biology of each is different, and so we need to take that into account to develop a cure. But having said that, I gave you the example a few minutes ago of acute leukemia with children and how we've gone from 10% to 90% cure rates in 50 years. So what defines cancers? There are two defining characteristics. One is that they accumulate aberrantly. So I'm going to use the words accumulate, grow, divide, proliferate interchangeably. And what that means simply is one cell becomes two, two becomes four, four becomes eight, and so on. So they grow or accumulate aberrantly, and they move. They move from where they originally started. And that movement or invasion is called metastasis. And if you break down the word metastasize, meta means beyond, and stasis is stillness. So it's double negative, beyond stillness, meaning it moves. So tumor is a derivative of a Latin word for swelling, and this means abnormal cell growth. So if you have a wart, for example, that's an abnormal cell growth. It's not a tumor. I mean, it's not a malignant tumor. It's not cancer, but it is a tumor because it is an abnormal cell growth. And to just give you some terminology, if you see a word that ends in the suffix oma, O-M-A, that denotes a solid tumor. So a tumor, again, is an abnormal cell growth, and it can be either benign or malignant. Benign is not cancerous, so the word tumor is not synonymous with cancer because a benign tumor is not cancerous. It's an abnormal growth, but it has not moved. In contrast, a malignant tumor or cancer is an abnormal cell growth that has invaded local or distant tissues. So here are some examples of tumor types. So benign, again, not cancer. It's a tumor because it's growing abnormally, but it has not invaded local or distant tissues. And an example of that is a lipoma. So OMA is a tumor. LIP stands for lipids or fats. So this is an abnormal growth of a fat-forming cell, and it's called lipoma. In contrast, if we look at a malignant tumor, which is cancer, so malignant tumor and cancer are synonymous terms. It's an abnormal growth that has invaded local or distant tissues. And an example of that is a melanoma. So OMA, again, is a tumor. The first part of that word, MELAN, stands for melanocyte. And this is a pigment-forming cell or a freckle. And melanomas are malignancies of freckles, essentially, that can spread throughout the body. And so this is a picture. But because biology is not binary, biology is always, not always, but it's usually a continuum, there are things that fall in between. So there are what we call pre-malignant tumors. These are ones that we know could become cancer if you let them grow, if you let them go long enough. So it's an abnormal growth, again, a tumor definition. It has not invaded local or distant tissues, but we know from experience that it has the potential to do that. And an example of that is a polyp. So this is a picture of polyps that you would see in the large intestine. And we know if left unattended or if not removed, these have the high potential to become malignant tumors and spread. So when you have a colonoscopy, this is what the GI doc is looking for. And then he or she can snip that out with scissors or some device at the end of the tube. Okay, so those are our definitions of tumors. So how do these things arise? I mentioned that rogue cells, cells that do not obey the rules, are the origins of cancer. And we have, the average adult has somewhere between 10 and 30 trillion cells in his or her body. So if one of those decides not to obey the rules, cancer can arise. So you can start to get an appreciation for why 40% of us will develop cancer in our lifetimes. So to go into this in a little more detail, we need to do some physiology and biology 101. So our bodies are comprised of systems, like the digestive system shown here. And the digestive system or systems in our body, let me get this pointer, are comprised of a variety of different organs, and each organ has a particular function. So the liver makes enzymes that break down the food. The gallbladder stores some of those enzymes and then injects it into the stomach where the food is. Pancreas makes insulin. The small and the large intestine take care of absorbing waters and nutrients and then excreting the waste. So this is a system, and we have about 13 of these in our body. And these are comprised of organs that have specialized functions. The organs are comprised of a number of tissues that have specialized functions again. So the stomach, for example, has muscle tissue and neuronal tissues, a variety of other types of tissues. And then tissues are comprised of cells of the same type. And this is the level that we want to look at. So this would be a smooth muscle cell that's in the stomach that's part of the digestive system. And again, if you want to go down even a more reductive approach, cells are comprised of molecules, which are comprised of atoms, right? But what we're going to focus on is the cell, because if one of these cells does not obey the rules, it can become malignant and lead to cancer. Okay. And so the information that cells follow are encoded in a molecule called DNA. And these are the instructions that cells have to perform their various functions. So DNA is an acronym for deoxyribonucleic acid. And this is a biochemical information storage and retrieval system. So DNA is comprised of only 4 molecules that are abbreviated by the letters A, C, G, and T. So everything that makes you up, makes you you, is the result of just 4 different molecules. Each cell in our body that has a nucleus, which is the majority of them except for red blood cells, has about 12.9 billion of these letters, which are called nucleotides, because they're in the nucleus. And the entire collection of DNA that a cell has is called its genome. OME is a suffix that's used to describe the entirety of something. So GEN is gene, which is a segment of DNA. So the genome is all of the DNA that we have. The proteome is all of the proteins we have. The microbiome are all of the microbial organisms that exist in our body. So OME is a term that's used widely in biology to denote the entirety of a particular biological species. And so analogies for this biochemical information storage and retrieval system include a hard drive on your computer, an owner's manual for a piece of equipment, or a cookbook. So you can use whatever one of these feels right for you. And in going down the hard drive and the cookbook analogy pathways, one file on your hard drive or one recipe in your cookbook is equivalent to 1 gene. And this is a unit of information that's used to produce a discrete product. So for a cookbook, one recipe would produce, let's say, lemon meringue pie. In our case, for biology, 1 unit of information or a gene is used to produce a discrete product, which is a protein. So a gene is a nucleic acid information. The protein is the molecule that actually does the work. So how do cells know what recipes to access in their repertoire in a given situation? So this is a cartoon depicting a cell. The red circle represents the membrane that keeps the outside out and the inside in. That satellite dish at the top represents what's called a receptor. And this is a molecule that can receive a signal and deliver that signal from the outside to the inside of the cell. The 2 structures within the cell are the nucleus that contains all of the DNA or cookbook, and the ribosome is a protein producing factory. So that's what's going to make the proteins that mediate whatever function the cell needs. So let's say you get a signal from the external environment to make cookies. So that signal is transmitted to the receptor, which receives it, and that delivers a signal, a biochemical signal, to the nucleus to tell it to open up the family cookbook and access that particular recipe. And then that recipe is copied. And then let's say, again, using the analogy, it's copied on a piece of paper that then is carried to the cook who is in the factory, the ribosome, that will make the gene product, which would be a protein or, again, a bunch of cookies. Because this information in the cookbook is so precious, it's retained within a protective organelle called the nucleus. So the DNA doesn't really get out of the nucleus. I'm not going to talk about mitochondria. That's an exception. In biology, there are almost always exceptions to the rule, so I try not to speak in absolutes. But by and large, the DNA stays in the nucleus, right? So it has to be copied and has to get out of the nucleus.
I'll get you, my pretty, and your little dog, too.
And so it's transcribed from the language of DNA to the language of RNA. So the NA stands for nucleic acids, but DNA differs slightly than RNA. And RNA is then copied, the recipe is copied into the language of RNA, it's transcribed into RNA. And then that RNA goes into the ribosomal factory that translates the language from RNA into the language of proteins. And so this is the process by which a cell can access its 25,000 different recipes in its cookbook. So I've mentioned numbers before, and that's leading up to this point, which is that the odds are not in our favor. So every day in our bodies, we make between 200 and 400 billion new cells every day. Every cell in our body contains 12.9 billion nucleotides, half of which were newly copied. So when one cell becomes two, its DNA is copied such that each of the daughter cells has the exact replicate as the parental cell. So half of this DNA is newly copied. And if you do the math, this works out to a trillion, almost 2 trillion billion letters of ACGT that are copied every day in our bodies. If you put it in a different context, if you were to lay this DNA out end to end in a single molecule thick string, it'd be about 200 miles of DNA, which is about twice the distance from the Earth to the sun. So this is how much DNA is being copied every day in our bodies. No copying mechanism is so precise that it will not make a mistake when you're dealing with numbers as big as 10 to the 21st per day. So mistakes are inherent, they're going to happen. And this is what leads to cancer. So cancers are genetic diseases that result from alterations, mistakes, mutations in the DNA sequence, and sometimes when they're expressed. So in that cartoon I showed you a couple of slides ago with the chef saying, make this recipe. If the chef said, make this recipe, but the cell misheard it, if you will, and made the wrong recipe, then pathology could ensue. So cancers result from the corruption or misuse of information, genetic information, which is DNA. The corruption results from changes in the DNA itself that can be very small, a single letter, or they can be large, where let's say half of the recipe is ripped out and half of another recipe is taped in its place. So those are large chromosomal abnormalities. The misuse results from changes in when and where and what genetic information is accessed. And this process is called epigenetics. The prefix epi means above. So oftentimes you hear that, this city was at the epicenter of the earthquake. The tectonic plates that caused the earthquake did not crash together within downtown LA. It happened 6 miles below the surface of where LA sits. So epi means above. So epigenetics means that the underlying nucleic acid is fine, but what's controlling when and where and what is used is not correct. And then finally, cancers are often genetically unstable, so they acquire mutations at a higher rate than their healthy, normal counterpart cells would. And this results from mistakes in the DNA copying or DNA repair machinery. And so when this becomes compromised, it can lead to greater accumulations of DNA mistakes or mutations at a higher rate that can occur when cells copy one another. So we have mutations or mistakes now. Well, not all of these are created equal. They fall in terms of cancer into two categories. One are neutral passenger mutations that are along for the ride. They merely accompany those functionally important driver mutations that can lead to cancer and are subjected to selective pressures. So it's this mixture of benign passenger and malignant driver mutations that together comprise the mutated gene sets of the tumors in question. So if you have a malignancy and the DNA of that malignancy is sequenced, you're going to get mutations in both passenger and driver genes. And the problem is that we don't know every driver gene. We don't know every passenger gene. So if the mutations occur in genes that we know of, and we know how they work, then we can act on that information, but otherwise we can't. So this is just a cartoon showing how this process occurs. So these driver mutations allow cells to overcome barriers to cancer. We have evolved a number of mechanisms at the biochemical and cellular levels that inhibit cancer, prevent it from happening, or remove malignant cells before they become problematic. And I've depicted these as these brick walls. So in the bottom right of the screen is an open green circle, and that's a normal cell. So let's say that cell begins to divide, and it acquires a couple of mutations. The gray mutation is a passenger mutation. It doesn't affect the biology of the cell, but the green mutation allows the cell to overcome that first barrier. And now it proliferates, and it starts to acquire more passenger and driver mutations. And the appropriate driver mutation here is depicted by that red horizontal or diagonal line. Now the cell keeps proliferating, acquiring more mutations until eventually it acquires enough mutations that it escapes all of these controlling mechanisms and becomes a clinically apparent tumor. This typically takes decades to occur, to acquire these mutations. I grew up on Long Island in New York. My dad loved to swim in the ocean, so we went to the beach every chance he could get. I grew up in an era before sunscreen was used widely. I'm fair-skinned. I got exposed to a lot of UV damaging, DNA damaging light as a kid. I've had several skin malignancies, early melanoma and a couple of other types of skin cancers that have been removed when I've been in my late 50s, early 60s. Probably the initiating mutation occurred when I was 4, 5, 6, 7 years old on the beach, but it just took decades for those other mutations to accumulate sufficiently for the cell to become fully malignant. And this is why trying to catch cancer early is critical, because it has acquired fewer mistakes, which means it's easier to treat. So this is a cartoon of colorectal cancer. And so stage 0 means that it's probably hard to see, but there's a little green bump here. This indicates that these cells are growing aberrantly. They're growing more than they should, but they haven't invaded any of the local or distant tissues. So this is a benign tumor. This would be, let's say, a polyp. But now at stage 1, you can see that it's starting to invade these local tissues. These double white lines are what are called basement membranes, and they separate the inside or the lumina of the intestine from the outside. But now you can see that the cells are starting to invade. So this would be a cancerous tumor because it's invading locally. And then there are different stages of invasion, 2, 3, 4. And again, this depicts this time-related accumulation of mutations. And so these polyps can easily be snipped out, and there's no worries because they haven't accumulated. But if you have a stage 3 or stage 4 cancer, these are starting to now access the superhighways of the body, which are the blood vessels, the vasculature, and the lymphatic vessels, which are part of our immune system. So if they can access a lymphatic vessel or a blood vessel, now they have access to the entire body, and they can spread and metastasize to distant organs like the liver. So now instead of trying to deal with one localized tumor, you're trying to deal with hundreds of different tumors that have disseminated to a variety of sites. So if you catch a malignancy early, it's much easier to treat than it is if it is caught later. So how do we get cancer? Age. Accumulation of DNA damaging mutations. And this depicts the frequency of cancer on the y-axis and age on the x-axis. Light blue are men, dark blue are women. And you can see that the frequency increases dramatically as an individual ages, simply to do this accumulation effect. This is about 10 years out of date. Now the ratio of men to women that get cancer is getting closer to 1. This increase in men versus women reflected the fact that back in the 1940s and 50s and 60s, more men were smoking cigarettes than women. So lung cancer accounts disproportionately for this difference. But now it's kind of evening out. The second way we can get cancer is just pure bad luck. You can inherit certain mutated genes that predispose you to cancer. During your normal everyday life, you can have a mistake in copying that is not corrected, or you can be accidentally exposed to DNA damaging agents in our environment, such as radon gas or ultraviolet light or something like that. The third way is through lifestyle. And here you're intentionally exposing yourself to things that we know can damage DNA. And DNA damaging agents are called carcinogens, things that can lead to cancer. And the poster child for this is obviously tobacco, cigarette smoking. So we've known for decades that smoking cigarettes can increase the probability of getting lung cancer. And if you smoke, you are intentionally exposing yourself to DNA damaging agents. When I speak to students and give them a similar talk and present this information, I tell them that of these three mechanisms for getting cancer, the only one you can control is your lifestyle. So if you minimize your exposure to things that we know damage DNA, you can decrease the likelihood that you will get cancer in sometimes in your life. Okay, so why is cancer sometimes deadly? One reason is that it compromises normal organ function. So these things grow more quickly than healthy cells, so they can starve, suffocate, and squeeze healthy cells and kill them that way. And an example of how this could be problematic would be if you have a kidney tumor. As that tumor grows, it's crushing the filtration units in your kidney and thereby compromising the removal of toxins, which is the normal physiological function of a kidney. So you're accumulating more toxins in your bloodstream than you would otherwise. Another way is that it can weaken the immune system, which can make you more susceptible to overwhelming systemic infections, which is a process called sepsis. Abnormal growths can block blood vessels or lymphatic vessels or digestive tract vessels. And so if you have a blockage, bad things can happen, such as bleeds or vascular problems such as aneurysms or strokes. They can cause chemical imbalances. So if you have bone cancer, the cancer cells are absorbing or breaking down the healthy bone cells, and that releases a lot of calcium into your body that you normally wouldn't have. If you have liver cancer, the liver is a site of making many, many, many different enzymes that are required for our digestive system, for example. So if you have liver cancer, you're getting perturbation of the balance of enzymes that's required for you to be healthy. And then there are substances called cachectin-like substances. And this is what tells the brain to stop eating when we feel full. But cachectin-like substances are not produced as well as they should be in cancer patients. And so a cancer patient probably is not hungry, both because they're feeling bad from the disease, they're feeling bad from the chemotherapy, but they're also getting these in their own natural substances telling them that they're not hungry. But the body still needs energy to function, so it starts to eat itself, if you will, where it feeds on normal tissues for energy. And this is due to a chemical imbalance. So the way I view cancers are like invasive species or weeds, for example, in this case. And this is a picture of a patient with melanoma. So again, this is a malignancy of a freckle. One freckle went rogue, and you can see how it spread throughout this person's leg. Unfortunately for this individual, it also spread to this person's brain. And these arrows depict those white dots, which are tumors that are growing. And as the tumors grow, they're compressing the healthy brain cells around them. And obviously, this can have lethal effects. So I think of cancers as an invasive species. Okay, with that background, so that's pretty much all I wanted to say about Cancer 101. Now I'm going to move into therapy. But before I do that, I'm going to take a quick break because my throat is getting dry. And I'm drinking soda, which is not good for me. So there you go. So the Masonic Cancer Center is where I work. I am a basic researcher. I have a PhD in immunology. I'm interested in using the immune system therapeutically to treat cancer. And I'll explain why in a few minutes. But the Masonic Cancer Center is one of only 51 elite cancer centers in the country that have this designation as comprehensive. And comprehensive means that we have research and clinical activities that span the entire cancer continuum, from how do you get cancer to how we can make an individual's life as comfortable and meaningful as possible when they have a terminal diagnosis of terminal cancer. And so we impact each of these areas. There are only 51 of these in the country out of over 1,500 cancer centers. So we're in the top 3% if you just do the math, but because we're in Minnesota and we're above average, I say our cancer center is in the top 1% in the country. I'm from New York originally, as I mentioned before. This is a jewel in Minnesota's crown. And New York has cancer centers as well, but Masonic Cancer Center is great. So let's talk about therapy and how it's evolved. So I mentioned before about Queen Atossa back in 400 BC. Surgery has been used for as long as we know to treat cancer. But as you can see, if it has metastasized, surgery is inadequate. In the 1900s, radiation and chemotherapy came into practice. And chemotherapy actually arose from the horrific effects of mustard gas on World War I soldiers. So after the war, scientists wanted to study how the mustard gas mediated its awful effects. And what they found was that these nitrogen-based gases formed chemical bonds with DNA in cells, and that caused the cell to commit suicide or die because its DNA was damaged beyond the point of no return. And then they made a leap of logic to say, well, we know that cancer cells are rapidly dividing, and if this nitrogen mustard gas kills rapidly dividing healthy cells, maybe it kills rapidly dividing malignant cells. And so that was what ushered in the chemotherapy era, which is still used as a foundational therapy for cancer today. And then radiation therapy delivers high energy to DNA that breaks it apart into little bits, and again, the cell commits suicide. There are problems with these approaches that I'll discuss in a moment, but you probably can guess what they are. But at the start of this last century, we've become more precise and more able to kill malignant cells and spare healthy cells. And the two general approaches for this type of targeted therapy are called precision therapy and immunotherapy. And what's been driving this evolution is specificity. Specificity is the holy grail of cancer therapy, meaning that if you can identify something that is unique to a malignant cell with respect to the healthy cell from which it was derived, and you can target that uniqueness and only kill the malignant cell and not any other cell, then you can deliver tons of the drug and not worry so much about the side effects that limit chemo and radiation therapy. So if we look at the evolution of cancer therapy, conventional therapy again is chemo and radiation therapy. These therapies kill rapidly dividing cells. And the reason they do that is depicted in this slide. So if you were to take the DNA in one of our cells and stretch it end to end in a single molecule thick string, it'd be about 7 feet long. And that 7 feet is wrapped up and packaged in a nucleus that's only about 1/5000th of an inch in diameter. So that's a heck of a packaging job. And the way that it's packaged is that it's rolled around protein balls that are rolled around themselves, that are rolled around themselves. And so you've got this tightly packed DNA that's in a chromosome that's essentially protein and DNA. For one cell to become two cells, though, it needs to unravel this DNA such that the DNA is accessible to the copying machinery of a cell, of the cell. So the DNA has to be unraveled and exposed, if you will, to the DNA copying machinery. And if a cell is rapidly dividing, at any given time, more of its DNA is going to be unraveled and exposed than a cell that is not rapidly dividing. And so if a cell that is rapidly dividing is treated with chemotherapy or hit with radiation, the tightly packaged DNA is not damaged, but the DNA that is exposed is damaged. And this is why rapidly dividing malignant cells and rapidly dividing healthy cells are killed by chemotherapy and radiation therapy. The next step in the evolution of cancer therapy was killing certain tumors preferentially. So as we learned more about the biology of tumors, we started to identify these unique characteristics. And then with that understanding, we could develop targeted therapies. An example of this is about 1/3 of metastatic colorectal cancers. Again, this is my cartoon of a cell. And about 1/3 of these cancers express this receptor for a growth factor called epidermal growth factor. EGFR is epidermal growth factor receptor. They express thousands to tens of thousands more copies of these receptors on their cell surface than a healthy colorectal cell does. So that means they're hypersensitive to very, very low levels of this growth factor that normally would not be recognized by a healthy cell. So that means that they get many of these growth signals that cause the abnormal growth of the tumor. So once researchers recognized this, we made reagents, the name of the most popular one is called Erbitux, that will block the receptor, and it prevents those signals from being delivered to the cell, so the cell becomes quiescent again. But the other thing that these drugs do is that they serve as red flags for the immune system. So these Erbitux-coated tumor cells are recognized readily by the immune system and then destroyed. But again, that type of drug doesn't work with every tumor, and it doesn't work with every type of metastatic colorectal tumor. The third step is where we're going for this patient-specific or precision medicine approach. And again, there are two avenues of study. One uses a combination of pharmacology, drugs, and genomics, and it's called pharmacogenomics. And these treatments are tailored to the genetic changes in both your healthy cells as well as your own cancer. And so what I have here is a depiction of 4 genetically distinct individuals that have the same type of cancer depicted by this open lightning bolt. So it's the same type of cancer, same mutations, but the individuals are genetically distinct. Their livers make slightly different enzymes. So let's say we have 4 drugs that we know can specifically target this disease, but some people have a strongly adverse effect to a couple of the drugs, where a couple of the others will work in them. So how do you know which drug to choose with which patient? So what we can do is we can sequence the healthy DNA in these individuals and identify what enzymes they make, and then correlate that to how the enzymes deal with the drugs. And then what we can do is we can pair the person with the appropriate drug. If this person on the right, the purple person, has a different disease, well, then we can give her a different type of drug that again is matched to his or her genetics. So this is where the field is going. We're at the field is in its infancy, so we're not able to do that with many cancers, but this is where it's going. The other area is the area that I work in, and that's using the immune system to treat cancer. So immunotherapy, as the name implies, uses the immune system to treat a disease like cancer. And it's been a game changer for cancer because of 4 characteristics of the immune system. One is specificity. I mentioned earlier that specificity is the hallmark of cancer therapy, is the holy grail, I should say, of cancer therapy. The immune system has evolved to be exquisitely specific. The immune system can distinguish between 2 proteins that are 99.99% identical. They may have 1 or 2 molecules that differ between them. The immune system can find that difference. It's incredibly potent. 1 killer white blood cell can kill thousands of tumor cells. It's long-lived. After these killer cells can no longer find tumors to kill, they either commit suicide themselves or they differentiate into a quiescent sleeper state, if you will, where they're long-lived memory cells that can become quickly reactivated if the tumor ever recurs. And they're adaptable. The immune system can change along with the changes in a tumor that it acquires during this unstable process that it goes through. So as subtle mutations occur within a tumor, the immune system can change along with it and adapt. It's almost like the evolution of a predator-prey relationship. The prey gets faster, the predator learns a way to get around that. And so for these 4 reasons, the immune system has an enormous potential to treat cancer. And we know from animal studies and anecdotal studies in humans that the immune system is constantly removing nascent or newly formed tumors every day in our body. So if you think back to that cartoon I showed you with the brick walls, I envision the immune system as being the last brick wall that a cancer has to overcome. And this process is called immune surveillance. So these different colored balls represent different white blood cells that recognize different molecules, and some of them will recognize the tumor as being foreign because it has acquired mutations that were not present in the DNA that we inherited. So the immune system says, hey, this is foreign, much like it would say, hey, this cell is infected with a virus and it can kill it. And we know that this happens every day. With that definition, clinically apparent tumors have escaped immune recognition. So figuratively, it's as if the tumor erected a wall between itself and the immune system that prevented the immune system from killing it and allowed the tumor to grow to become clinically apparent. So even though immunologists have recognized those 4 characteristics, specificity, potency, longevity, adaptability, as being really attractive to treat cancer, we haven't, until the last 10, 15 years, been able to exploit those 4 characteristics to treat cancer because we did not know how this wall was built. We didn't know how the tumors were evading immune recognition. But due to basic research in immunology and tumor biology, we're starting to learn what the bricks and the mortar are in this wall. And with that knowledge, we've been able to overcome this barrier. So one way that we can overcome it is through a process called immune checkpoint blockade. And figuratively, what we're doing here is we're lowering the height of the wall, or we're destroying the integrity of the wall, such that the immune system is no longer suppressed and no longer has can access the tumor and kill it. And Jimmy Carter, who is unfortunately in hospice care now, was diagnosed when he was in his early 80s, mid 80s, with a type of skin cancer, melanoma. And it was stage 4, it was terminal, but he was treated with this immune-based therapy and it allowed him to live for another 10 years, where he still continued to build houses for Habitat for Humanity. The other strategy is the one that my colleagues and I are using here at the U. And that's where we can engineer these immune cells. So again, these colors represent different things that they see. It's great that we have this variability because that means we can respond to almost anything that nature throws at us because we'll have a cell that can recognize it. But the frequency of any one of these cells that is specific for a tumor antigen or a tumor molecule is really, really low. And so it can be blocked by this wall. What we can do, though, is we can take these white blood cells out of an individual, genetically reprogram them to see what we want them to see, and this overrides what they were born to see. And then we can grow them up into vast numbers, billions of numbers in the laboratory, and give them back to a patient. And so now this swamps the tumor-induced immune suppression wall like a tsunami breaching a seawall. There are too many killers to be suppressed. And this strategy is called, one type of this strategy is called CAR, which stands for chimeric antigen receptor. I'm not going to get into that. But I just want to show you this video on how these killer cells work. This is a pretty cool video. So I'm going to stop and then share this, if I can find it. Here it is. Okay. Is this YouTube video visible?
All of us lurks a serial killer.
Yep, we can see and hear
A
it.
killer whose primary
Thank you.
function is to kill and then kill again. These are cytotoxic T cells, a specialized member of our white blood cells. They patrol our bodies, identifying and destroying virally infected and cancer cells. And they do so with remarkable precision and efficiency. There are about 5 million T cells in a teaspoon of our blood, engaged in the ferocious and unrelenting battle to keep us healthy. These amorphous blobs move around quite rapidly, pushing out their leading edge and probing their environment as they go. When a cytotoxic T cell finds a cancer cell, membrane protrusions rapidly explore the surface of the cell, checking for the telltale signs of cancer. They kill their targets using poisonous proteins visible here in red. These cytotoxic granules move down special pathways in the cell called microtubules to the interface between the T cell and the cancer cell. The T cell punctures the surface of the cancer cell and delivers its deadly cytotoxins. This is very important in our bodies, where cells are packed together, as it focuses the lethal hit on the target and minimizes collateral damage to the neighboring healthy cells. The fate of the cancer cell is sealed. The T cell then moves on, hungry to find another victim.
I get a kick out of that. I'd like to know if I put a stethoscope up to my arm, if I'd hear that music, but I don't think so. Okay. So here's how this CAR T cell therapy works. Oh, before I go into this, so in that video, there was a section where one of the green killer cells was kind of looking over the surface of the blue cancer cell, and we call that interrogation. If the T cell didn't see anything on the surface of the cancer cell that looked foreign to it, it would detach and go off and find another cell to interrogate. So what these CAR T cells do is that we have engineered those green killer cells with a molecule that will definitely find something on the surface of the cancer cell. So instead of maybe 1 in 10 million of your healthy white blood cells being able to recognize this mutated protein, 100% of them that we put in do. And so the way that this works is that this is a patient. We will remove the blood from the patient, isolate these killer cells, genetically modify them in the laboratory by putting in this CAR, which is again is chimeric antigen receptor. And then we grow them up to billions, literally, and then put them back into the patient where they can then go and find the tumor cells and kill them. To give you an idea as to the market size of this, it's thought to grow to, it's going to be in the hundreds of billions of dollars. So the CAR T cell therapy market size is predicted to hit 10.5 billion roughly in 2027. And it's growing at an annual compound average growth rate of almost 50%. So it's skyrocketing. So there's huge interest in this from pharmaceutical companies as well as academic institutions. The cost of one of these things, it's not cheap. It's on the order of high 300s to high 400,000s per infusion. And this excludes what the hospital charges and what the physician charges and everything else. So you're looking at $500,000. But typically, you only need one infusion because you're injecting your own cells. So your body is saying, yeah, okay, we know you. We're not going to reject you. And these cells live in your body for as long as we've looked, which is out to 12 years now. So it's a living drug. Many commercial health insurance plans pay for this type of therapy, but not all. Medicare covers it. Medicaid coverage varies depending upon the state in which you live. These therapies, though, are given to patients that have failed the standard chemotherapeutic regimen. Okay. So the take-home messages, I'm just ready to finish up. I think this is not my penultimate slide, pretty close to the end. Cancer is an umbrella term that describes hundreds of diseases. No, because I'm giving a talk.
Okay, he has the space reserved. That's what we were saying. He has the space reserved.
Okay, 1 second, then I'm almost finished. Okay,
thank you.
thanks. Cancer is an umbrella term that describes hundreds of diseases. They grow uncontrollably and metastasize or spread. They're heterogeneous because they result from the corruption of misuse of genetic DNA. And combinations of precision targeted therapies are the future for cancer patients. And comprehensive cancer centers like our cancer center are ones that you should exploit. So let's get back to Emily. Emily was treated with CAR T-cell therapy. She was the first pediatric patient treated. And again, this is just how the therapy works. In May, she'll be 18 years old. She'll be cancer-free for 11 years. So her family has allowed us to use her name and likeness to promote this type of immunotherapy. So with that, I'm going to stop. And I've been just told I have to leave this room, even though I was told I could have the room. So I am going to move to another space and I will be available very, very shortly. Okay, Laura, I'm back.
You're back. Do you need a minute to
Nope, I'm squared away.
catch your breath? Okay.
Yeah.
Okay. So are these is this CAR T cell therapy still a phase 3, or is this
No,
standard treatment?
there are 4, actually, there are 6 current FDA approved therapies for this. So it's not in clinical trials anymore. It's available.
Okay, why can't these things be used right away? Why do people have to go through chemotherapy first before they can do this?
Yeah, that's a good question. Because we didn't know how these things would work at first. And they're still kind of an unknown, even though they're FDA approved. Some patients won't respond, some patients will respond. And so I don't know if this is true or not, but my gut is that it's a legal issue, because if you have a strategy that's been used for 50 years and you get, 60% cure rate, and you bypass that and you go directly to this and it doesn't work, you may get sued for malpractice if you didn't follow the standard procedure. So I don't know the exact answer, but that's my gut feeling.
Okay. What kind of so you mentioned a success rate. I maybe just threw that out, but it probably depends on different cancers. But what is the general success rate?
Yeah, so it's interesting. So the success rate, you're exactly right, does vary from cancer to cancer. To give you a ballpark estimate, 50 to 60% of patients are long-term survivors, and that's defined by 5 years or more. So, and again, remember, these are patients that have failed all of the other therapies before. So that number is phenomenal. But again, it's not 100%. And the story with Emily is fascinating because she was the first pediatric patient that was treated with CAR T-cell therapy back in she was diagnosed in 2010 and then treated in 2012 with CAR T-cell therapies. And Carl June, the fellow, the physician that treated her in Pennsylvania, in Philadelphia, said that if she had died, the whole field would have come to a screeching halt. And the reason is that for a pediatric clinical trial, if the first child on that trial dies, nobody wants their child to be child number 2, right? So even though this therapy doesn't cure 100% of the patients, he said that they got really lucky that Emily was one of those that it did work for, because otherwise all of these other patients wouldn't have had that benefit. So, yeah.
Any idea why this therapy doesn't work for all patients?
Yeah, so that's an area that we're actively, when I say we, I mean the whole field, is actively looking at. And there are a number of reasons for it. One is that the product, what we're doing is we're taking out your white blood cells, modifying them, manipulating them in the lab, and then giving them back to you. If you've already gone through a lot of therapies and you've had the disease for a while, your white blood cells may be not great, right? And so the starting material is suboptimal. That's one reason. Another reason is that for some reasons that we're just beginning to understand, sometimes when you put these cells back in the body, they become what we call exhausted, meaning that they're there, but they're not killing the cancer cells. And we don't know exactly why that happens. We think it may be related to how we treat them in the laboratory before we give them back to you, or what the composition of the cells were in the beginning. So maybe most of your cells are exhausted to begin with, and we didn't know that, and we gave them to. So now we're trying to figure out, okay, should we take just this particular type of white blood cell and get rid of the rest, because we know that's going to work better. So these are some of the things that we're doing. And then it's disease specific. So about 15% of all cancers are what are called liquid tumors. And those are white blood cell tumors that are floating around in your body, right? And those are more easily treated in general than many solid tumors, because the liquid tumors are accessible to anything you put in the bloodstream. There are no barriers. Whereas solid tumors, particularly pancreatic cancer, literally build protein walls around them that make it very difficult for drugs to diffuse through and get into them. So liquid tumors have had the most success with these CAR T-cell therapies. And in fact, I don't think that there are any FDA-approved CAR T-cell therapies for solid tumors. The 6 or 7 that I know of are all for liquid tumors because of that accessibility issue.
Both leukemias and lymphomas? Or
Yeah, so lymphomas are considered liquid tumors, even though they're solid masses, but they're in the lymphatics and they're accessible to things that we put into the bloodstream so again, biology is not binary. So liquid would be a leukemia floating around in your bloodstream. A solid tumor would be something like pancreatic. And you can think of lymphomas, which are white blood cells, but those are growing in your lymph nodes, as being kind of in between the two. But we still consider them liquid because they're not as solid as a solid tumor.
Yeah. So are there side effects to the patient getting this immunotherapy?
Yep, there sure are.
Ugh.
Yeah, so nothing comes without a cost. And there are both short and long-term problems with this therapy. The short-term problem is overwhelming inflammation. So inflammation is useful if it's localized. So if you get a bee sting, you get a red welt and your skin you'll get a bump and it'll be hot. That's because locally there's more, the blood vessels become leaky, and that allows white blood cells from your bloodstream to get into the tissue to take care of whatever caused the problem. But if you have systemic inflammation throughout your whole body, your lungs can fill with fluids, your brain can fill with fluid and then compress the brain and cause problems. So systemic body-wide inflammation is not good. And this is what happens when you put in 1 billion of these killers that can find 1 billion target cells in their body. You're going to get widespread inflammation. And so that killed a couple of patients in the very early clinical trials until we learned how to handle it. So now we have a couple of drugs that we can dampen that down with. So that's an acute toxicity. Another acute toxicity, in fact, this is what I'm working on, is neurotoxicity. So for reasons that we still don't know, many of these therapies can cause neuronal problems. And this was completely unexpected because the animal studies and everything we did in the lab before we went into the patients didn't suggest that this would happen. So what my lab is doing is we've developed a mouse model for this CAR T-cell therapy that captures all of the good and all of the bad we know happens in people. And the bad includes this neurotoxicity. So what we're doing now is to try to understand how it happens with the long-term goal of preventing it. So systemic inflammation, neurotoxicity are 2 acute toxicities. And neurotoxicity is particularly problematic because it can span a spectrum of presentations such as mild disorientation to cerebral edema and death, which means swelling of the brain. But even in its mildest forms, if you're disoriented, you can't be discharged from the hospital because you may walk in front of a bus, or you don't know what you're doing. So there's a cost to that. And then the long-term cost is immunosuppression. And so I haven't said what the molecule is that these CAR T cells see. And what it is it's a molecule expressed on only a type of white blood cell that can become malignant, but it's also expressed on the healthy cells as well. So when we put these killers in, they're wiping out half of your immune system. And so these patients like Emily are immunocompromised for life. That seems horrible, but the flip side is that she would have died when she was, 7 years old. So it's that. So what happens in research is that what is available in the clinic is about 10 or 15 years behind what we can do in the laboratory. And so what we've done once we recognize that problem is we've developed suicide switches, and we've developed all of these other strategies that will allow you to get rid of the CAR T cells once the tumor is killed entirely. And then your immune system will rebound from precursor cells that are in your bone marrow. So that's where the field is going. But again, there's a 10-year lag between what we can do in the lab and what is FDA approved, because during that 10 years, it has to go all through these clinical trials to make sure it's safe and effective, and what dose do we give and that sort of thing. So again, to summarize, short-term acute toxicities include this massive cytokine tsunami. Cytokine is a pro-inflammatory molecule. It's this tsunami that breaks over your entire body, and neurotoxicity are the short-term problems. The major long-term problem is immunocompromised, because your B cells, which is the type of cell, is being killed.
Maybe these also are reasons why it's not a first-line therapy. Don't
Yeah.
know. Just wondering. Yeah.
Yep. Yeah, that's a good point, Laura.
Yeah. How many patients, if you know, how many patients have been treated here at the U with this therapy?
Yeah, I don't know the number off the top of my head, but it's approaching hundreds.
Mm.
So, and we're learning more and more so clinicians. I have my office right next door to a couple of people that actually treat patients. I just deal with mice. They don't let me near people, which is smart. But we talk to each other, and so that's how we developed this mouse model. Because I have a colleague who's a pediatric oncologist, and he does bone marrow transplantation and he does immune cell transplantation, and so the two of us together developed this model. And so I'm dealing with the basic biology of it, and then hopefully he can bring that into the clinic.
How many other centers in the US are using this therapy?
There are many centers in the US that are using it, because now that it is FDA approved, it's not restricted to just research centers like the University of Minnesota or MD Anderson or places like that. So it's becoming more and more widespread. And what is done is that not every center can make these CAR T cells. So what is done is that your blood is taken out in your local clinic, and then it's shipped off to a company, or typically a company that will do all of the modifications. And then they send those cells back to your physician, who then infuses it into you. So you can imagine, for that process, it's pretty complex, which is why one of the reasons why it costs as much as it does. And so not everybody can do that. And we can do that here. Other people can do it, like at MD Anderson or Sloan Kettering, they can do it there. But it's become so widespread because of this ability to take out the blood, ship it, have it made, shipped back, and then infused on site.
Okay, how does this differ from a stem cell transplant?
Yeah, so it differs in the type of cell that is given. So they're both transplants, meaning that you're taking something and you're giving it to somebody else. So that's the definition of a transplant. But I'm going to digress for just a second. And so the short answer to your question is there are different cell types. For CAR T cells, we're taking an immune cell that's ready to go and kill. For a stem cell, it's like a baby cell that has to go to school to learn what it's going to do. So that's the short answer. The reason that stem cell transplants are given is because not enough of the chemotherapeutic drug can be given to kill the tumor without killing the patient. And one of the fastest dividing populations of cells that we have are cells in our bone marrow that make the blood. Red blood cells and white blood cells. These are constantly being made in the hundreds of millions every day in our body. And so the cells that make them are dividing and dividing and dividing, and their DNA is exposed. And so when you give chemotherapy, it's killing those blood forming cells. And if you give enough of the drug to kill the tumor, you're also wiping out somebody's blood system. So with a transplant, what happens is that they give the patient an incredibly high dose of chemotherapy that is hopefully high enough to wipe out all of the tumor cells, but the toxic side effect is that it's going to wipe out their blood system, and then they would die. So to prevent that from happening, several days after the drug is given and it's been cleared from the body, you're given these stem cells that replace the ones that were killed in your bone marrow. And then they give rise to all of the white blood cells, there are many different types of white blood cells. So that's what is a stem cell transplant. Stem means it can give rise to pretty much anything. What we're doing is we're taking one of the branches of that stem, if you will, that are mediating the function that we want, which is to kill. Not every white blood cell will kill. So we're taking the ones that kill, we're modifying them to see what we want them to see, and then we're giving them back. So we're not destroying your immune cell, your immune system entirely. Your bone marrow is still okay. But so that's the difference.
Okay, thank you. Are there any age limits to this type of therapy?
Probably, but I don't know of them. The younger you are, the better it's going to work. But it's certainly given to elderly individuals, and I'm not going to define what elderly is, but it probably depends upon the health of the individual. So if the individual is in poor health in their 50s, probably not a good candidate. If they're in great health in their 80s, it may be a good candidate. Again, I'm not a clinician, so I don't know the exact answer, but it's certainly used for pediatric patients. So, the lower end of the spectrum is, 3, 4, 5 years old. The upper end, I think, depends upon the health of the individual. But one example, let me step back for a bit. One of the CAR T cell therapies that's approved is for multiple myeloma, and that typically predominates in elderly individuals. And so they are, they're available. They can be treated with this type of therapy.
Okay, so you explained why CAR T therapy works best in the liquid cancers is accessibility. They can get at them.
Yeah.
But for solid tumors, because lots of people are asking questions about solid tumors, do solid tumor cancer cells also express unusual receptors so that
Yes.
eventually there might be a therapy for this?
Yes, they definitely do, and that's where the majority of the research is occurring, because 85% of all cancers that we get in the country are solid tumors. And so it's a multi-step process to develop this type of reagent. The key is to identify what do you want to target. And ideally it would be what's called a neo, meaning new, a neo target that's derived from a mutation in that tumor, because then that mutation would not be expressed in any other cell because it's unique to the tumor. So many people are looking for these neo markers. They're hard to find. So what people do is they try to compromise for a tumor associated marker that's expressed at a higher density or maybe only on the tumor cell and very few other cells. So one of the big problems with the solid tumors is identifying the target that you want to choose, right? What are you going to choose? The second problem is getting the cells in there. So cells get into tumors through the blood vessels. And tumors themselves, once a solid tumor gets bigger than the point of a pencil, it has to make its own blood supply to get enough food and oxygen to survive. But the blood vessels it makes are not as good as healthy blood vessels. And so for even if a white blood cell is in the tumor vasculature, which is a name for the blood vessels in there, it has a harder time getting out than it would if it was in my arm or a healthy organ. So the tumor vasculature presents a bit of a barrier. Then the other thing is, I mentioned that wall that is formed by tumors, that can turn off these killer cells. So the tumor microenvironment, the environment that the tumor makes around itself, is naturally immunosuppressive. So we really have to ramp up these killer cells to overcome that immune suppression. And then finally, some tumors like pancreatic cancer literally have walls around them of protein that make it very difficult for the tumor cells to get in there. So these are the barriers. What are you going to target? How are you going to prevent the cells from getting turned off when they get there? How are you going to get them there? How are you going to get around the problems with the blood vasculature? These are the barriers that we're trying to overcome to extend this therapy to solid tumor therapy. But having said all that, there are certainly clinical trials that are ongoing now that show some success with certain solid tumors. So people smarter than me are figuring out how to get around these issues.
Okay, so also a lot of questions about melanoma, which I know is another solid tumor, but I know that some patients have been effectively treated with immunotherapy, which is maybe monoclonal antibodies. I'm not sure.
Yes, yes.
How does CAR T therapy differ from monoclonal antibody?
Yeah, so both of those are types of immunotherapy, but antibodies are proteins that are secreted by certain types of white blood cells. The antibody producing cells are called B cells because they come from the bone marrow. So those are protein molecules that are secreted. They're obviously a lot smaller than a cell. So CAR T cell therapy by definition is a cell. Antibodies are protein molecules. And the advantage of using an antibody is that antibodies are exquisitely specific. So I've got a water bottle here, right? So if my hand is an antibody, I'm trying to get it in focus, not working, but you get the idea. So let's say I have a monoclonal antibody. It can bind to this cell, this thing really, really tightly. If I have a different type of water bottle that's bigger, it won't bind that well. So antibodies are incredibly specific. And so we can exploit that specificity to do pretty much whatever we want. And the way that the monoclonal, mono means one, clone means progeny from a single cell. So monoclonal antibodies are proteins that are all derived from an original progenitor cell. And that means that all of those antibodies are identical to one another. That means it's essentially a chemically defined molecule because they're all identical to one another. And that makes it much easier for us to manipulate and test and that sort of thing. So we can make monoclonal antibodies that bind to molecules incredibly tightly. They'll never let them go. And I mentioned that wall before, that is a suppressive barrier that is made between a tumor and the immune system. Examples of that wall are molecules that are on the surface of a tumor cell. So if a white blood cell comes along, it will get basically the brakes applied to it and it will turn off and become inactive. But what we can do is we can make an antibody that binds really tightly to that brake on the surface of the tumor, such that when the white blood cell comes, it's not getting that turn off signal. It's not getting that inhibitory signal because it's blocked by that antibody. And that's what the immune checkpoint blockade therapy is. The immune checkpoints are the brakes that are on the surface of a tumor. Blockade means we're blocking the brakes. So you're removing the inhibition, and now you're allowing the immune cell to kill, to do what it's supposed to do, and it's not being turned off. So it's an immune molecule. It's made by an immune cell, but it's a molecule. It's not a cell itself. And pretty much every combination that you can think of is being applied here. So we're making molecules that bridge a tumor cell with a killer cell. We're making molecules that basically put the step on the gas as well as blocking the brake. So all these different combinations are being tested in the lab.
Do you think the cost of this therapy will become cheaper over time?
Yeah, I do. Yeah, as more and so there's the mass action, right? As more of it's made, it'll become less expensive because manufacturing procedures will improve. One of the big pushes is to develop what are called off-the-shelf reagents, meaning that we won't have to take my blood out. We can go to the freezer and pick out a cell that's frozen that we know is going to work in me and then grow it up and give it to me. And so that will cut down on cost and on time. So yeah, it definitely will decrease in cost. I don't know when.
Yeah. Okay, switching tracks now. Do you know anything about this purported efficacy of the polio virus for glioblastoma brain tumors?
Yeah, so that type of, so I'll give you more jargon. There's something called oncolytic. Onco means cancer, oncology. Lytic means it lyses. So there are some viruses, when they get into a cell, will burst the cell open, and that's how they release all of their thousands of viral particles that the cell makes, right? But what people here at the U and other places are doing is making oncolytic viral therapy, meaning that they'll take a virus, tweak it such that it will preferentially infect a cancer cell. And then when it gets in the cancer cell, it will lyse it. And that does two things. One, it kills the cancer cell that it infected. And the other thing is that when it lyses that cell, that sends off signals to the immune system that something bad has happened. And it's basically like raising the red flags. So it elicits an anti-tumor response at the same time it's killing the cell. And we used to think that the brain was what's called an immunologically privileged site, meaning that there was no immune function going on in there. We know that that's not the case now. And so there are you don't want to have inflammation in the brain. So how the immune system works in the central nervous system is different and not nearly as well as understood as it is in the periphery and in other parts of the body. But if you can preferentially kill a tumor cell and at the same time release all of these what are called danger molecules that alert the immune system to something going on, you can elicit an immune response. And I think that that's that type of oncolytic viral therapy is being used to treat a number of tumors. I'm not 100% sure if the polio virus strategy is that, but I think it has something to do with it.
Okay, thank you.
Yeah.
So someone else here is asking, they knew someone with melanoma years ago. Something was taken from this patient's lymph nodes, and then the physicians made a serum of some sort and then re-injected the serum, and the patient was cured. Do you know what that might have, what therapy?
Yeah. So melanoma is an interesting disease. It's a favorite of tumor immunologists because it's infiltrated by a lot of white blood cells. And so what I think happened in this example is that there are naturally occurring tumor-specific white blood cells that were elicited by the melanoma. And lymph nodes are filtration units in your body, and they're also town hall meeting places, if you will, for white blood cells, where they can go and find things that get them excited, and then they can expand. So when I was a little kid, if I got sick, my mom would feel my glands and say, "Oh, your glands are swollen. Yeah, you're sick." And I didn't know what that meant until I became an immunologist. And your glands here, these are called cervical lymph nodes. And what happens is that if a white blood cell gets into the lymph node, and again, the lymph node is a filtration unit. So if this is a bacteria coming in, it gets caught in the lymph node, and then an immune cell comes through and it sees it and goes, "Oh, I know this," and then it can expand. That's what causes your lymph nodes to expand and swell, or your glands to get swollen, because you're getting all of these white blood cells that are growing, and then they're released into the body where they can take care of whatever caused the infection to begin with. So what I think happened in this case is that white blood cells specific for the tumor were growing in the closest filtration unit to the melanoma. And I'm going like this because there are lymph nodes in your armpits called axial lymph nodes. So if I had a melanoma here, some of those white blood cells would be in my lymph node, and that's an enriched source of those cells. So they could take that out and grow them up to large numbers in the laboratory and give them back. Very similar to what's done with the CAR cells, but they were not genetically manipulated. They were just enriched and grown in the lab and given back to you.
Can you expand on how that, is it a T cell? Yeah, T cell, how that T cell is modified to become that CAR? How is the recognition protein added? Is it a CRISPR method?
That's how it's going right now. But again, 10, 15 years ago, it was through viruses. So there are viruses that package genetic information, and viruses are very good about getting their genetic information into a cell. So we've used viruses that don't cause disease, that don't make more copies of themselves, but we use them as delivery vehicles to give genes to a cell. So your healthy T cells would be infected with one of these viruses that encoded the information for this CAR. And then the genetic information would be incorporated into the DNA of the T cell. And so every time the T cell divided, it would copy that gene that made this protein on the cell surface. So initially they were introduced via a couple of different types of viruses. Now there are electromechanical ways of getting DNA in. And then, as you just mentioned, Laura, CRISPR-Cas9 is being used to put the DNA in exactly where we want it to go, where we think it will do the best.
Okay, can these CAR T cells themselves become cancerous?
Yeah, it's an interesting question. That was that was the biggest fear that the field had, because we couldn't control where the virus integrated that CAR gene. And if it put it next to a cancer-causing gene, it might turn on that cancer-causing gene. So that was a big concern. And that's the advantage of using this CRISPR technology, because you can define exactly where you want that gene to be inserted. So there are things called genetic safe havens, where you can put in anything and it's not going to affect what's near it, because it's a safe harbor, if you will. But that did not happen. But what did happen one time, though, so there's no example that's been reported of one of these CAR T cells becoming cancerous itself. But what did happen in one case report was that when they purified the T cells, a few of the leukemia cells were co-purified as well. And then they were reinfused back into the person. And they now had this CAR on there that basically let them go crazy, and they killed the patient. So you got to be really careful. That's why, Walgreens Pharmacy can't do this, because it's pretty complex. So that's the only example that I know of where one of these products that was made in the laboratory actually had a cancerous effect.
Wow. Well,
Yeah.
we certainly appreciate your efforts at trying to solve this problem. Obviously, this will keep you busy for the rest of your career.
Yeah, it's Laura, I feel like I'm getting paid to do my hobby. It's just a lot of fun. And one of the most important things that we do is community engagement like this. So I very much appreciate the invitation. And if folks have different community organizations that they would like somebody from the Cancer Center to come and speak to them about, we're happy to do that. So, just shoot me an email and we'll work it out.
Wow, that would be fantastic. All right, so we're sadly out of time at this point. Thank you again, Dr. Pennell, for all of your information. Of course, the audience, fantastic questions, and the OLLI program. Many of you have also written asking for the web link to the YouTube channel where you can rewatch this fascinating talk. So if you have a piece of paper nearby, it's youtube.com, that's YOUTUBE.com/@OLLI_UMN. And if you didn't catch all that, you can just go to the YouTube channel, just type in youtube.com. And then when you get there, type in the search box, OLLI UMN, and you'll find us there. So again, thank you, everyone. Please join us next week at the same time for the presentation, Bug Appetit, Edible Insects from Western Taboo Food to a Healthy, Eco-Friendly Diet for the Future, with Dr. Sujaya Rao, who's professor and head of the Department of Entomology here at the U. Take care. We'll see you next week. Bye-bye.